Hello, I'm Cameron Burke, and welcome to this Kaplan MCAT Science Review for the Adaptive Immune System. This video covers the major landmarks you might be asked about on test day relating to the adaptive immune system, including the adaptive immune system in general, B cells and antibodies, and their counterparts T cells and cytotoxic immunity, as well as activation, recognition, and immunization. By the end of this review, you should have a good idea of the parts of the specific immune system and how they work. If you achieve this goal, you should be able to answer questions like this one about bone marrow's function. You'll probably recall from your previous studying that in general, immunity is the ability of an organism to resist disease, and there are two types of immunity. The first type is innate immunity, which doesn't target specific pathogens, but that's not the focus of this particular review, so we'll set that aside for now. The star of the show for this review is adaptive immunity, or the specific responses the immune system can throw at recognized pathogens. There are two types of adaptive immune responses. The first is humoral, which involves antibodies moving through liquid tissues without the involvement of cells. And the second is cell-mediated response. The exact type of response varies depending on the pathogen present, but we're going to review the broad categories of adaptive responses you could be asked about when you take the MCAT. Some of the most well-publicized characters in adaptive immune defense are the B cells and T cells. B cells are in charge of antibodies, which are the main player in humoral response, while T cells play a larger role in cell-mediated response. Both B cells and T cells are initially born in the bone marrow, but in the case of B cells, they stay in the bone marrow while they mature into adult cells. They're then activated in the spleen and lymph nodes. On the other hand, T cells move from the bone marrow while they're immature and then develop to maturity in the thymus. Recall from a moment ago that B cells produce antibodies. Antibodies are one of the most important factors for humoral immunity. Each pathogen has its own antigens, or foreign proteins. B cells make antibodies that specifically respond to those antigens on foreign cells. Antibodies can either be stuck on the surface of a cell or just generally released from lymphocytes near the area where an antigen is, but the result is the same. Antibodies act like a flag or beacon for one specific pathogen, so more aggressive immune-related cells can track it down. Then the invaders can be recognized and destroyed, and you can stay healthy. Once antibodies have been released, they can only bind to one specific type of pathogen. If an antibody tries to attack a pathogen it's not compatible with, it won't be able to bind and serve as a signal to the rest of the immune system. There are three ways antibodies can get their job done. The first is opsonization, which is when antibodies stick onto leukocytes through receptors for the constant regions of antibodies, which lets the leukocytes know they need to come and eat the pathogen. The second way is for antibodies to force pathogens to get stuck together in a giant pathogen ball, stopping the pathogens from reproducing and allowing them to be phagocytized. Finally, antibodies can surround a pathogen to stop it from getting into tissue, which doesn't kill the pathogen, but it does make it so the pathogen can't infect new cells, and that's basically just as good. In addition to floating around in bodily fluids, antibodies that were created during prior infections also exist on the surface of healthy host cells. One way to activate the adaptive immune system is to have binding occur between the familiar antigenic proteins and their matching antibodies on the surface of a cell, which causes B cells to be activated, which triggers the creation of plasma cells and memory B cells, which can both produce antibodies for a pathogen. Remember what mast cells are for. They cause an inflammatory response to allergens. When antigens happen to bind onto the antibodies of mast cells, degranulation occurs, which causes the granule contents in the cell to be released, triggering an inflammatory response. If you're an immunologist, you would need to know a lot of information about the structure of antibodies, but because we're just reviewing what you need to know for the MCAT, we're only going to get into a couple of areas that give antibodies their unique identities depending on what pathogen they're for. Antibodies are made of chains of amino acids. Each individual antibody has two identical copies of a heavy chain, which is different from all other antibodies. The heavy chain is a more dense region coded for more genes than the light chain. Each antibody also has two identical light chains of amino acids, which are less dense than the heavy chain regions, but equally important for creating a variable identity for the antibody. Together, the heavy and light chains combine at the tips of antibodies to create the antigen binding region at the end of the variable region, so named because it's the variable or the differing factor in each different antibody. The heavy and light chains are attached to the constant piece of the antibody, or the piece that's the same for all of the types of antibodies, through disulfide links and non-covalent bonding. As you might imagine, while the constant region of the antibody is important, the variable regions made up of the heavy and light chains are really the star of the show. Antibodies have specific polypeptide sequences in their variable regions that correspond directly to one particular type of antigen. Because B cells are in charge of quickly activating antibodies to best match invading antigens, they hypermutate. Hypermutation lets the cell try many iterations of the variable region until it lands on the one that can most effectively bind to the antigen. Once hypermutation has done its job and a good match has been found, clonal selection for only that type of cell happens. Clonal selection allows for only the effective types of B cells and T cells that can actually bind the pathogen to be activated, and the rest of the cells that didn't quite get the right polypeptide sequence remain inactive. The variable region's an important part of the antibody that's responsible for sticking to the antigens, but the constant region has a really important job too. The constant region is responsible for binding to the immune cells and letting them know it's time for them to get to work. The constant region is a part of every antibody, and it's also recognized by many of the most important innate immune cells, including natural killer cells, macrophages, monocytes, and eosinophils. While this list isn't exclusive, it is a good representation of some of the most important cells to know for the MCAT. Unlike innate cells, B cells are specialists. Each only makes one type of antibody, and that's it. Fortunately, healthy immune systems have a ton of B cells, so many types of antibodies can be produced in response to many types of antigens. While each antibody is different, they do come in one of 5 categories of isotype, including D, E, G, A, and M. Depending on which isotype an antibody is, it has a different shape. D, E, and G are monomers, while A is a dimer. M is a pentamer, which can be recalled on test day by remembering that M stands for many. In addition to having different shapes, the isotypes each have jobs they are more or less suited for, so they get used in different circumstances. Sometimes B cells change the isotype they produce when signaling proteins called cytokines indicate that conditions have changed, to allow another isotype to be more effective. B cells are generally responsible for the production of antibodies, which is definitely important to know. But on test day, you could also be responsible for knowing the subtypes of B cells, because they take on more than one form. The first of these forms is the naive B cell. Naive B cells are little baby B cells who've never encountered an antigen. Once they first get exposed to a particular antigen they can respond to, the primary response from the immune system is initiated. B cells for that particular antigen start proliferating after the start of the primary response, and this is when antibody production starts slowly happening. This is firstly accomplished by quickly producing plasma cells, which have short lives dedicated entirely to producing antibodies, and by creating memory B cells, which can respond to the same antigen later if they encounter it again. Memory B cells come into play during secondary response. Primary response takes about a week to get going, but the second time your immune system senses a familiar infection, the memory B cells launch a much faster secondary response. The secondary response, as compared to the primary response, is super quick. The memory B cells left over from the first infection stick around in the lymph nodes. When they detect a familiar pathogen, the memory B cells get straight to producing antibodies without messing with the plasma cells. Unlike the first time, when naive little baby B cells encountered a strange virus and had to make plasma cells and then develop into memory cells and whatnot to ultimately start a slow response to produce antibodies. The secondary response antibody production from the memory cells when they encounter the same virus is much faster. B cells have more or less one main task, and that's to produce antibodies. That said, once the antibodies are stuck on the antigens, you need some other cells to actually respond to that signal, and that's where T cells come in. Before that point, T cells grow to maturity in the thymus. T cells can mature in 2 ways. The first way is positive selection, where only the cells who can appropriately respond to the antigen pieces presented on the surface of attacked cells are picked to finish growing up. The second type of maturation is determined by negative selection, which is when overeager T cells that attack body proteins are forced to apoptose, leaving behind only the cells who can respond to pathogen proteins. Thymosin helps T cells mature no matter which type of selection they underwent. Once thymosin's been applied and the T cells are physically all grown up, they get to leave the thymus. When they actually meet an antigen, they undergo clonal selection just like B cells do, but this can only happen once they've been exposed to an antigen, not when they're still naive cells. When T cells grow up, they differentiate into several individual types of T cells, which you could be asked about on test day. The nicest of these are helper T cells, which are basically the coordinators of a total immune response. Helper T cells release lymphokines in the face of infection, which lets all the other immune cells know that they need to get on the scene. There are many immune-compromising conditions, including HIV and AIDS, that prevent helper T cells from turning on the rest of the immune system. Those particular diseases highlight just how important helper T cells are for immunity. Helper T cells know there's an infection by the antigens that are stuck on MHC class II cells, which if you'll recall are on immune-specific cells. There are two types of helper T cells, each of which do an important job in terms of activating the rest of the immune system. Th1 cells release interferon gamma to activate macrophages, which starts them eating bacteria, and Th2 cells turn on B cells to produce antibodies. If helper T cells are the nice organizers that say, okay, immune system, we need to get to work, then cytotoxic T cells are your immune system's trained assassins. Their main job is to kill infected cells. Like helper T cells, cytotoxic T cells recognize antigens found in major histocompatibility complex, but that's about where the similarities end in terms of their tasks. Cytotoxic T cells recognize antigens on MHC class I cells, which are not immune specific. These are just cells that do other jobs in your body that have been infected. When that happens, the cytotoxic T cells poison the infection-presenting cell, and then it dies. And that's the end of the virus in that cell. So far we've reviewed nice helper T cells, which tell the immune system to get to work, and merciless killer cytotoxic T cells. But they also have a third counterpart who's basically middle management, the suppressor or regulatory T cells. These guys make sure the immune response isn't too weak or too strong, but just right. If the immune response is too weak, infections will kill you. But if your immune response is too strong, your own immune system will harm your body, which is what's going on in autoimmune disease. So regulatory T cells make sure your immune system's strong enough to kill infection, but not so strong that it kills you too. They do this by turning off self-reactive lymphocytes through destroying them, or repurposing them to suppressor cells. Structurally, suppressor cells are similar to helper T cells, but they have an additional protein called FOXP3. On test day, the presence of FOXP3 proteins help you decide if you're looking at a suppressor T cell or a helper T cell. So keep that in mind for lymphocyte questions. In the same way that you have memory B cells for fast antibody response during reinfection, you also have memory T cells. These are activated every time a pathogen tries to infect a host after the first time. Just like memory B cells, they can respond much more swiftly than naive T cells can during an initial infection. But the end result is the same. They kill the infection. This particular review focuses on the adaptive immune system, but any truly effective immune response relies on the cooperation between both the innate and the adaptive systems. There are several categories of infectious pathogens your body could encounter, including bacteria, viruses, fungi, parasites, and prions. Depending on the particular pathogen, the immune system will react in different ways. For example, a macrophage from the innate immune system would be fine to kill a rogue bacterium that entered the bloodstream through a cut. But you're going to need the adaptive immune system to prevent you from catching last season's flu virus a second time. While the adaptive and innate immune systems usually work together to keep you healthy, sometimes the immune system as a whole gets a little overexcited about its job, and this results in hypersensitivity. Hypersensitivity includes autoimmune disease, which happens when the immune system just starts specifically responding to certain types of tissues without effectively determining if an antigen is from you or from a germ. It also includes allergies, which is when the immune system gets a little crazy about killing everything that isn't you, including things that don't actually hurt you, like kitten hair proteins and flower pollen proteins. These are not exactly an emergency, but the immune system acts like it is. This is probably going to seem like a lot of words that you're supposed to read and keep up with, but don't get stressed out about it, because it's actually the exact same process we just reviewed, but with harmless allergens instead of pathogens. The immune system's process for dealing with allergens is exactly like its process for dealing with actual pathogens. This starts with B cells recognizing the allergen protein, and plasma cells releasing antibodies. The antibodies activate mast cells, which specialize in allergic reactions, to get them ready for the next time they encounter allergy proteins so they can recognize them. When that happens again, mast cells release histamine and cytokines, which then make you feel sick. In the ideal healthy immune system, T cells and B cells that would react to good self-antigens and non-dangerous environmental proteins are downregulated, so usually hypersensitivity isn't a problem. One of the best things about the immune system, despite its occasional failure to recognize that dust is not lethal, is its ability to create immunity once it's been exposed to pathogens. Immunity to a pathogen can result in a couple different ways. The first is our current subject, adaptive immunity, which you have to develop. And the second is innate immunity, which comes standard with every healthy immune system. Adaptive immunity can come to you through natural and artificial means. In general, natural exposure means catching an illness, and vaccines are considered to be an artificial exposure. But you end up with immune memory either way. In a vaccine, instead of catching a live microbe that wants to use your body to reproduce, you just get dead or weakened microbes. or even just proteins present in microbes. And your immune system develops secondary immunity before it faces the real disease. Then if you encounter the real thing, your immune system's ready and you don't get sick. Some particularly famous examples are smallpox vaccines and the polio vaccine, but you've probably received many other types of vaccines yourself. In active immunity, such as vaccines or experiencing disease, your immune system has to do the work to stop you from getting sick. In passive immunity, someone else's immune system does the work and you just get to benefit. Some examples of passive immunity include maternal antibodies, such as those transferred through breast milk. But it also includes just about any method where antibodies are artificially transferred into an individual after being produced elsewhere. The bad news about passive immunity is that it's temporary, because only antibodies are being introduced, not immune cells. So there isn't a team of cells waiting to produce more antibodies if they're needed. That said, there are several ways to transfer antibodies, which can be especially useful to improve immunity in immunocompromised people like infants or cancer patients. These include but are not limited to placental transfer, breast milk, and straight-up intravenous transfers. Now that you've made it through the content part of this review, you can test out your learning goal progress with these practice questions. The first one deals with antibody binding. The correct answer is A. The variable region is the part of the antibody that can recognize specific antigens. The second practice question tests you on your lymphokine knowledge. The right answer to this question is D. Helper T cells primarily secrete lymphokines to get the rest of the immune cells on board with the immune response actions. You might be asked about improving immunity on test day, and that's also what you're being asked about in the third practice question. If you chose D, you're right. Sanitation, like washing your hands or refrigerating your food, is really important to prevent infections from happening in the first place, but if you get sick, cleaning everything around you will definitely not help your body get rid of pathogens. Finally, check out what you know about hypersensitivity. B is the right answer. Hypersensitivity can be limited by killing defective immune cells, and overenthusiastic T cells are negatively selected for in the thymus. To recap, lymphocyte precursors in bone marrow turn into T and B lymphocytes. B cells produce memory B cells and plasma cells, which confer humoral immunity through 5 individual antibody isotopes. T lymphocytes proliferate into helper T cells, which activate other immune cells, suppressor cells, which keep immune response levels at appropriate strength, and cytotoxic T cells to kill other cells who have been infected. They also create memory T cells, which trigger a fast cell-mediated response the next time an infection hits. Additionally, the immune system as a whole has 2 branches. This includes adaptive immunity and innate immunity. Adaptive immunity can be natural or artificial. Natural immunity includes passive antibodies received from a maternal source, and active immunity resulting from infection. Artificial immunity includes passive immunity from antibody transfers and active immunity from vaccinations. And that's the end of the adaptive immune system review. Thank you so much for watching this video. Hopefully it'll help you if you get asked about the adaptive immune system when you take the MCAT.